Welcome to the fourth lecture of week 12. So, in this lecture, I am going to discuss immunoassay techniques. So, this is part of our various biophysical and biochemical methods that we are studying. So, today I am going to introduce what are immunoassay techniques. So, I will give you a general overview. Then I will discuss these specific methodologies: ELISA, Western blot, immunoprecipitation or co-immunoprecipitation, immunohistochemical assay, radioimmunoassay and immunofluorescence assay. So, you will see that the basic principle for all these assays are very similar, and apart from this there are also many other methods which will be some variations of these methods. So, what are immunoassay techniques? Immunoassay techniques are highly sensitive biochemical tests that utilize the specificity of antigen-antibody interactions to detect and quantify the proteins in a sample. So, these techniques use the high specific and highly tight interaction between an antigen and an antibody. So, in theory we can raise very specific antibodies against any antigen, right? And this quality of antibodies are used in this technique. So, immunoassay techniques are widely used in clinical diagnostics, research and pharmaceutical development. We will see examples as we go through. So, here are the important immunoassay techniques. The first one is radioimmunoassay. In this case, the antibody or the antigen are radio labeled, and since we are using radioactivity, one has to be very careful in terms of using this assay. However, the advantage of this assay is that it is highly sensitive. Very small amount of antigens can be detected using radioimmunoassay. The next one, enzyme linked immunosorbent assay or ELISA. This is the most popular one. So, in this case, the antibody is linked with an enzyme and it is the enzyme activity that is used to detect. Okay. So, since this is an enzyme activity, it will amplify your signal and it also increases the sensitivity. So, these are all variants of these methods. Chemiluminescent assay. So, in this case, chemiluminescence is used for detection. In fluorescent immunoassays, you use fluorescent labels for detection. And then there is lateral flow immunoassay. So, in this case, it is a simple strip based detection system. Examples of this will be pregnancy test, right? Okay. So, what are the applications? They have applications in clinical diagnostics, in drug development, in food safety, in environmental monitoring, right? So, in clinical diagnostics, it can be used to detect various infectious diseases such as HIV, hepatitis. It can be used to measure different hormone levels such as insulin or thyroid hormones from blood samples. It can be used to identify cancer biomarkers, right? For example, PSA for prostate cancer, right? So, there are many examples like this. Drug development, of course, these are very important for drug development and they are used in very basic research in different laboratories. So, they can be used for high throughput screening of different drug molecules, or they can also be used for immunogenicity testing. So, in the food industry, they are also very useful. So, they can be used to detect allergens and contaminants that can be present in a food that is mass produced. Environmental monitoring, so it can be used for detecting toxins and pollutants. So, let us look at enzyme linked immunosorbent assay, which is most probably the most popular technique in immunoassay techniques. So, ELISA is a highly sensitive and versatile immunological assay and it is used to measure antibodies, antigens, proteins and glycoproteins in blood sample, right. So, you can measure any of these things using ELISA. What is the principle? It relies on the use of enzyme conjugated antibody, which means that the antibody on the heavy chain covalently linked enzyme is present. For example, let us say HRP, horseradish peroxidase, right, and its reaction will be used for detection. Now, it can be so this enzyme is linked to the antigen or the antibody or the antigen to detect the presence of a target molecule, which is antigen or antibody. So, typically you will detect antigen using the antibody. What is the readout? The readout is a color change is produced when a substrate is added to the enzyme, right, indicating the presence of the target. And again, the applications are in diagnostics. So, various biomarkers for cancer, in case of autoimmune disease, so biomarkers for autoimmune disease, biomarkers for metabolic disorders, diagnosis of infectious diseases, pregnancy test, etc. Right? it can be used for vaccine development and drug testing, and it can be used for food quality analysis and detection of toxins. Now, ELISA comes in different flavors, right? So, the first one is direct ELISA. So, in this case, it involves the binding of the primary antibody directly to the antigen. So, this is the antigen. So, AG is antigen, right? So, the antigen is immobilized. Now, you add the antibody which specifically binds this antigen, and then here you have some enzyme that is linked. So, once this binding happens, then you have to of course wash off so that any non-specific interaction is gone, and then you pass the substrate. So, this substrate will get converted to product. There will be some color change which you can detect. So, this is direct ELISA because your antibody is directly binding to the antigen. Next one is indirect ELISA. So, in this case the antibody, so there are two antibodies, there is a primary antibody and there is a secondary antibody, right. So, the primary antibody binds to the antigen coated on the plate. So, again the antigen is here on the plate, the primary antibody which is raised against this binds to this. Now, there is a secondary antibody to which the enzyme is linked, and this antibody binds to the primary antibody. So, this antibody is raised against this primary antibody, right. So, now you can use this. So, then again you add the substrate and from the reaction you can detect it, right. Now, the advantage for this ELISA over this ELISA is that here the enzyme linked secondary antibody can be constant and this can vary. So, you can raise different primary antibodies against different antigens and use the same enzyme linked secondary antibody which will detect this, and you can have your ELISA. Okay. In this case, you have to link your enzyme to the primary antibody every time. Okay. So, this turns out to be cheaper than this. The third one is sandwich ELISA. So, in this case, it uses two antibodies for the same antigen. Okay. So, the first one is a capture antibody. So, here the antigen is not on the surface, it is the capture antibody that is on the surface, and your antigen, it is most probably in some liquid sample, let us say. So, you flow it, your antigen will bind to this capture antibody, right. So, this is practically more useful, and then you use a detection antibody which is this green one, right. So, this will bind to the antigen. The only catch here is that in this case these two antibodies should identify two different epitopes of this antigen. So, the capture antibody will identify an epitope which is on this side, and this antibody will identify an epitope on the other side. Now, once this antibody is there, the secondary antibody will bind to this antibody, right, the detection antibody, and then you can do your reaction to detect, right. And finally, the last one is competitive ELISA. So, it involves the competition between the antigen in the sample and a labeled antigen for binding to a specific antibody. The amount of signal inversely correlates with the antigen concentration in the sample. So, this is more for a quantitative determination of how much antigen is present in a particular sample, right. So, in this case, you have the antigen from the sample which is there, and you have this other antigen. Okay. So, this you have, so you will compete this off, and when this compete this off, your antibody comes off, right. And then, so basically the signal, more signal that you get means more of this is coming off, right. So, that is why it is inversely correlated to the antigen concentration in the sample. Okay. So, this is the basic process. So, that antigen is coated on the well. In the second one, we saw that there will be the capture antibody which will be coated on this well. Now, you have to add some sort of blocking agent so that you can reduce non-specific interaction, right. So, some people use BSA. So, there are different mixtures that people use, right. And then you add your primary antibody which will bind to this antigen very specifically. Then again you have to wash with different buffer conditions so that you remove anything that is non-specifically bound, and then you go for the detection. So, you add the secondary antibody, again wash off anything that is non-specifically bound, and then you add your substrate to get your chemiluminescent signal, and then you can detect it using some detector. Okay. So, another very important and very robust and highly used experiment is Western blotting. So what is Western blotting? It is a biophysical technique that allows for the detection and quantification of specific proteins in a mixture, right? So it uses the principles of gel electrophoresis for protein separation with antibody based detection. So let's see. Western blot works by separating proteins based on size through SDS page. We have already seen SDS page. Let's see, you have a cell lysate, right? So you have a mixture of proteins. You can separate them out through SDS page. But the problem is that you don't know whether the protein that you are looking for, whether it is present in this mixture of proteins or not. If you have purified your protein, there is only one band, then that's not a problem, right? But if it's still a cell lysate, it's a mixture of let's say several thousand proteins, then you don't know whether your protein is present. And even if you know the mass of your protein, let's say it is 25 kilodalton, there might be multiple proteins which are present at that position, right? So you don't know for sure whether your protein is present in that particular band which is near 25 kilodalton. Okay, so that is where we use detection using an antibody. To do that, what is done is all these proteins are transferred. So the separated proteins are transferred to a membrane, and then the specific protein is detected using labeled antibodies. So applications: protein detection and quantification. So you can detect and you can also quantify using densitometric analysis. So validation of antibodies, it can be used to confirm the specificity of antibodies against a target protein. So if you are developing some antibodies, you can use that. You can use Western blot to validate that, or you can use it for actual research purposes. So post-translational modifications, for example, phosphorylation, glycosylation, ubiquitination. So one can raise antibodies against these post-translational modifications of a particular protein, and then use that to detect how much of the protein in a cell in a particular condition is phosphorylated or glycosylated or ubiquitinated, right? And it can be also used for detection of biomarkers for various diseases. Okay, so this is the basic principle, right? So you have a cell lysate. It can be from bacteria, it can be from mammalian cell, right? And then we run an SDS page gel electrophoresis. So you have these separated proteins. Now these proteins will be transferred into a membrane. Typically PVDF membrane is used, right? So for that we will use again electrotransfer. So we will use a gradient of electric field and we will transfer it. Now one thing that you have to be careful about is that transfer of these proteins is not exactly the same. So larger proteins will transfer at a different rate than smaller proteins. So one has to be aware about the mass of the protein that you want to look at, right? And then adjust your transfer conditions accordingly. So once it is transferred to this membrane, then you have your protein on the membrane. So you add the specific antibody which will bind to that, right? And for detection, you add the secondary antibody which will bind to the specific or the primary antibody, and this will have some enzyme linked. So again, this is similar to ELISA, right? And then you add your substrate, you get chemiluminescent signal, and then you use some instrument to capture that signal, right? So you use chemiluminescence imaging. So standard gel dock apparatus are present, are available to do this. Okay. So next is immunoprecipitation assay. So this is a versatile biophysical technique used to isolate and study specific proteins from a mixture such as cell lysates or tissue extracts using an antibody specific to the target protein. So for example, there is a specific protein and you want to know that whether another protein, so protein A is there, you want to know whether protein B binds to it or not, or in some cases you might not know what are the other proteins that binds to protein A. So you want to identify those proteins, right? So in that case, immunoprecipitation assay is very useful. And there are various types of immunoprecipitation assays to study protein-protein interactions. So this will be called co-immunoprecipitation, or protein DNA interactions, and this will be called chromatin immunoprecipitation or CHIP. And then you can also use to detect post-translational modifications in proteins. So the principle involves capturing a target protein, right? So let's say protein A, which binds to other proteins and forms a complex. So we will have an antibody which will bind to target A, the target protein A, and then that antibody is actually conjugated to a bead, so that we can then spin down that bead. So the bead will precipitate, the antibody will precipitate, and the antibody will bind to your target protein A, that will also precipitate, and the target protein A, if it binds to other proteins, they will also precipitate. So you can isolate all of this together, and then you run a gel, use Western blot analysis, use other techniques to identify which are the other proteins that are binding to the target protein A. Application: study dynamic changes in protein expression profiles, right? You can detect post-translational modifications, you can isolate and study protein binding partners, and you can study changes in protein interactions or modifications in response to drug treatments or in response to different disease conditions, right? So you can look at control cells versus disease cells and see how these interactions change. Okay, so this is the schematic of the experiment. So let's say we have a cell lysate, we have protein of interest, and now we add our antibody, right? So this antibody will bind specifically to our protein, let's say that is protein B, okay? And then we add these beads, so these beads will bind to the antibodies, and then we can spin it down and we will isolate our protein, this blue, from the rest, and then we can run it on a gel and do Western blot and other methods to detect it specifically, right? So to do co-immunoprecipitation, let's say there are this mixture of proteins and this blue protein binds to this green protein, right? But there is this yellow and red, this all other proteins are there. We don't know that this green protein binds to this blue protein. So we want to see whether something binds to this target protein, and if it is, what is that other protein, right? So you have the cell lysate, you add your antibody, it binds to it like so, and then you add your beads. So you have this capture complex, right? So the beads, antibody, protein blue, and then this green protein is bound to this blue protein. So now you can wash and elute, or you can spin and wash, so you can use all these different methods so that these rest of the proteins are gone and you are left with this complex. And then you can run it, so denature everything. So if you denature it, these will all come off and you can run it on a gel, and then you can detect this protein blue and this protein green using specific antibodies raised against them. If you don't know what is protein green, you can cut out the band that is there and you can send it for mass spec analysis, right? So there are various methods by which you can identify what are the proteins which are binding to your target protein, which is this protein blue. Immunohistochemistry assay, right? So it is an immunological technique for detecting the presence of specific proteins within tissue sections using antibodies. So far what we have done is we are looking at cells, we are breaking up the cells, looking at cell lysate, right? So these are more in vitro experiments. But what if you want to see things in more biological environment? So for example, a tissue section, right? So in that case, immunohistochemical assays are very useful. So the principle is, again, a primary antibody binds specifically to a target antigen inside the tissue. The antigen antibody complex is visualized using an enzyme conjugated secondary antibody or a fluorophore that generates a detectable signal, right? So your detection can be colorimetric or it can be a fluorescent signal. The resultant signal reveals the distribution and localization of protein of interest within the tissue. Okay. So if you have a mixture of cells, you can see which cell types have your protein of interest, or within a cell, you can use confocal microscopy to see whether it is localized in a particular organelle inside the cell, right? For example, whether it is in the nucleus or whether it is in the cytoplasm or whether it is in the mitochondria, you can do that. Okay. So again, the applications are very similar. You can study dynamic changes in protein expression profiles. You can detect biomarkers for various diseases and disorders. You can use it for high throughput screening of drugs, and you can study interaction between different cell populations. So this is how it looks. So let's say you have this organ, and then there are instruments like microtome which can be used to create very thin slices of this tissue, right? So this is your tissue layer, and then you go through the same steps. So add primary antibody, there is a secondary antibody which binds to the primary antibody, and then you can use chemiluminescence, or this secondary antibody can have a fluorescence molecule attached to it. So, you can see it as fluorescence or you can see it as a chemiluminescence, right, and then you see it under a microscope. So, there is something that is very similar to this also. In this case, radio immunoassay, RIA, is again a highly sensitive in vitro assay technique that is used to measure minute concentrations of antigen, right. So, in this case, we are using radio labeled antibodies instead of enzyme conjugated antibodies, and since we are using radio labeled antibodies, the signal you can detect very low signal. So, the sensitivity is very high. Okay. So, it is based on the competition between radio labeled and unlabeled antigens for a limited number of antibody binding sites, right. So, when a known quantity of radio labeled antigen is mixed with a specific antibody, the unlabeled antigen in the sample competes with the radio labeled antigen for the binding sites or the paratopes. So, the paratopes are shown here, right, on the antibody. The unlabeled antigens replace the labeled antigens that are already linked with the antibodies. This increases the amount of free radio labeled antigens in the solution, right, and you measure that free radio labeled antigens, and from there you get some amount. So, you can quantify it. Okay. So, here is your antibody, radio labeled antigens are added, you have the complex. Now, you add your sample which has the unlabeled antigen. Okay. So, this unlabeled antigen will replace this radio labeled antigens. So, now you have this free radio labeled antigens. So, you quantify those, and from there you get a measure of how much unlabeled antigen is present, because this free antigen will be directly proportional to the free unlabeled antigen that is present in the actual patient sample. Okay. So, again it can be used to study protein localization and trafficking, visualize cellular organelles such as mitochondria and nuclei, detecting immune markers on cells, diagnosing different diseases like autoimmune disease, identify cancer specific markers in tissues or cells, studying the expression and localization of tumors, etc. Okay. So, the last one is immunofluorescence assay, and this is something that we have already seen, right. So, in this case the antibody is conjugated to the specific fluorescent level, and there are all these different fluorescent labels like DAPI, FITC, PE, and this one, and you can see they are different colors, which means that they fluoresce at different wavelengths, right. So, we can use a combination of these different markers to look at different things at the same time. So, that is the advantage of immunofluorescence assay. So, again you will have your cell or tissue samples, and then you will just add your antibody, right, and then look at it under microscope. So, we have already seen this, that we are using this antigen antibody specific interactions. So, there are all these different fluorescent molecules. So, that is the excitation wavelengths are shown on the left and the detection emission wavelength are shown on the right. Okay. So, we can, for the same sample, we can use different detections. So, something that looks at the nuclei, something that binds to the microtubule, which is then using seen using green fluorescent, Golgi seen using yellow, actin fibers seen using red, and mitochondria seen using magenta, right. So, the same cell you are looking at these different components using different antibodies conjugated to different fluorescent dyes, and you can combine this image to get an image like this, which shows the localization of all these different molecules inside the cell. You can see the actins are on the mostly on the cell membranes, microtubules are spread throughout, that is the nucleus, right, and mitochondria is also distributed throughout the cell. So, to get an image like this, of course, you have to use confocal microscopy. So, this is something that I have already discussed in earlier lecture. So, you can go through these books for the use them as a reference for the topics that I have covered in this lecture. Thank you.